Welcome back to our series of short lectures devoted to statistics for proteomics. The topic of discussion today is going to be focused on estimating the significance of differential protein abundance. And the next time we are going to discuss a related topic, which is the significance of functional enrichment of differentially abundant proteins. To begin with, it's important to acknowledge that there are very many software packages that you can use to estimate significance of differential protein abundance simply by providing estimates of peptides or protein levels and getting an output of estimated p-values and false discovery rates. My aim is not to discuss the practicalities of these methods or to compare them. Rather, I want to discuss the intellectual foundations, the principles that go into computing p-values and false discovery rates so that you understand how you can evaluate these different software packages, how to choose the appropriate package for your analysis, and importantly, how to write your own pipeline when the packages available do not provide the necessary flexibility for the analysis that you want to perform for the data that you are analyzing. Any method for estimating differential protein abundance is dependent on these three factors that I have enumerated here. They're of pivotal importance, and I'm going to elaborate on each one of them in the subsequent few slides. The first foundational pillar in estimating differential protein abundance is to know the degree of dependence or independence in the data points that you use to estimate differential protein abundance. It is essential to realize that biological replicates do not necessarily provide independent data points. There are many sources of dependence due to systematic artifacts that can occur again and again in different measurements. And below here, I have summarized a few examples of such systematic errors, which include, for example, ionization biases of different peptides. This bias is particularly relevant when we perform the estimates using estimates of absolute protein abundance, because the absolute protein abundance in one set of conditions can use one set of peptides, in another set of conditions can use another set of peptides, and differences in the inherent ionization efficiencies of those different peptides might provide a systematic difference that is being interpreted as differential protein abundance, while in fact, we simply have differential peptide detection and peptide ionization. Another example is ionization suppression, which is particularly relevant to label-free approaches that cannot control for such ionization suppression. And to be more explicit about it, an example of ionization suppression is the suppressed ionization of less abundant peptides by an abundant peptide that is coeluting and might be present in different amounts in different types of samples. And therefore, we measure different intensities for the less abundant peptides in large part because of the variability of the abundant peptide suppressing their ionization rather than because of differential abundance of the corresponding proteins. Another example is interferences. When fragments that belong to one peptide are being used to quantify another peptide and to estimate its abundance. And this is relevant to data-independent acquisition methods, relevant to other types of analysis as well. There are clever approaches that can help control such interferences, but nonetheless, there always remains a potential that they're not completely corrected and they can lead to systematic biases. Another very well acknowledged and widely discussed source of systematic errors is the co-isolation and co-fragmentation of precursors in the context of isobaric mass tags. This is very widely acknowledged and an important caveat to keep in mind when one uses TMT, iTrack, and other similar approaches. Another source of systematic errors can be the difference in biochemical fractions obtained from various isolation methods. For example, when we fractionate different cellular parts, nuclei, mitochondria, endoplasmic reticulum, cellular membranes, and so on, these fractions may contain different buffers that can contribute to systematic biases in estimating the protein abundances in those fractions. And there are very many other possible systematic errors that are not included in this list. This is just a short sampling to emphasize the need to consider the possibility that your measurements are not independent. So how can you get closer to having independent measurements, or to phrase it otherwise, how can you have less dependent measurements? A commonly used approach to that is to have replicates. And what I would recommend in the context of this approach is to try to change those replicates as much as you can. For example, by changing the measurement method, you can use both in one set of measurements, data-dependent acquisition, in another set of measurements, data-independent acquisition. You can use MS1-based methods such as SILOC and label-free analysis, or you can use MS2-based methods such as isobaric mass tags. And these different methods are going to have their own systematic artifacts, but because they're somewhat method-specific, by changing the method and the experiments, you reduce the degree of dependence of the measurements that are used. Another approach is to use different peptides originating from the same protein as providing relatively independent estimates for the relative changes of that in the abundance of that protein. And this approach can be quite powerful because different peptides have different physical and chemical properties. They elute during different times of the chromatography. They're affected by different artifacts. And its main limitation is the fact that those different peptides might be shared in a different way with different proteoforms. And therefore, we have to account to the extent we can, as best we can, for the existence of proteoforms when you use different peptides to quantify a protein. And that is going to be the subject of a future lecture of this series. Now, another fundamental pillar of estimating differential protein abundance is being able to estimate the errors in the data. Some of those errors are going to be systematic and they can be corrected, as I'll elaborate on the subsequent slide. And some of these errors are random, and we simply have to estimate them as best as we can in a way that is specific to the to each protein and each peptide, as I'll discuss further on. Here is an example of a commonly observed systematic bias, systematic error that correlates to the abundance of the proteins or messenger RNAs. It's an artifact that is very commonly observed in DNA microarrays, also observed in RNA sequencing data, where the relative changes that are being estimated for a particular gene are dependent on the abundance of the gene, as you can see on the left, with the inverted U-shape dependence. And in such a case, we don't expect to have the dependence, and we can subtract it from the data. We can first estimate the dependence, for example, by fitting a lowest to the data, and then subtracting the value of the lowest from each corresponding data point, we can obtain the corrected data shown on the right, where the data have been centered around 0 and do not show such a strong bias. This correction itself may introduce biases, and therefore one has to be cognizant of that and careful to evaluate the degree to which conclusions at the end of the study depend on having performed that correction and having performed it in a different way. So this is not a panacea, but this is certainly one approach that when used in a controlled fashion can help remove some systematic biases. Another example for a systematic bias that is commonly removed from proteomics data are biases that depend on the retention time of peptides. We can have the relative ratios of peptides be a function on the retention time, the order in which peptides elute from the chromatographic column. And because we don't expect that this necessarily should be the case, it is sometimes corrected. And the same measure of moderation and caution should go into performing corrections of biases correlated to retention times. Of course, there are also random errors, and these are much easier to handle statistically because simply by averaging measurements performed multiple times, we can reduce them, and it is easier to handle them in terms of established existing statistical models. One very important aspect of random errors to keep in mind is that usually those errors depend strongly on the abundance of the molecules that we measure, and this is true for messenger RNAs measured by DNA microarrays, RNA sequencing. It's equally true for peptides and proteins measured by mass spectrometry and by different approaches as well. It's a very general phenomenon where the errors for less abundant proteins are larger and the errors for more abundant proteins are smaller. If we don't take into account this dependence, we are going to underestimate the significance of differential protein abundance for abundant proteins. And more problematically, we are going to overestimate the significance for the abundance changes of lowly abundant proteins. And therefore, we can simply take into account the random errors distribution for each peptide relative to its intensity and prevent such systematic errors in the estimates of statistical significance. And this brings me to models that we can use for estimating differential protein abundance and the corresponding assumptions. The models can be categorized into two broad groups. The first group is parametric models. Generally, parametric models make more assumptions, but they also provide more statistical power when those assumptions are fulfilled. Despite having more assumptions, these models are oftentimes the right tool to use if we have only a couple of replicates. And because the non-parametric models will not have sufficient data to identify differential abundance in most cases where it does exist. Examples of such models include linear and polynomial models. They can be particularly appropriate when we have dose response type of data or time series data. The familiar t-test, which we can use if we are comparing only two conditions, and its generalization to the analysis of variance when we have many more conditions. And again, when these approaches are being used, one should be aware that the p-values are highly dependent on the assumptions being made and the degree to which those assumptions are satisfied by the data. When one has sufficient data, one can use a more data-driven approach that makes fewer assumptions by using non-parametric methods. They have less statistical power, which can be compensated for by having more data, and make fewer assumptions, which is good. These methods can include, for example, the rank sum test, which is a non-parametric counterpart to the t-test. It can include Spearman correlation. It can include a non-parametric ANOVA, which is ANOVA performed on the ranks of the data points rather than the data points directly, and so on. Having done the differential protein analysis, you should be aware that there are two types of measures that one can use to quantify the confidence in differential protein abundance. One type of measures is focused on evaluating the confidence in individual proteins, and such measures include p-values and posterior error probabilities. They're not the same, they're distinct, but both p-values and posterior error probabilities reflect the degree of confidence for the differential abundance of a particular protein. In contrast, the false discovery rate is an example of a measure that applies to a subset of proteins. It does not give you the confidence in the differential abundance of a particular protein, rather it gives you the confidence for an entire set of proteins. And in that context, it is important to realize that a set of proteins that is defined as differentially abundant at a very low false discovery rate may contain proteins that are certainly not differentially abundant. The example to the right is one shows you how that can be possible. If we have hundreds of proteins that have very low posterior error probability, we can have a few proteins, in this case indicated with a red bar, that are certainly not differentially abundant, but nonetheless a set of proteins as a whole has 1% false discovery rate. And the same can apply even if you have 0.1% or 0.001%, you can still have within a large set of proteins, a few proteins that are not differentially abundant. And therefore, if the interest is in a particular protein, one should not use the false discovery rate of the entire set as a confidence measure for that protein. An important caveat to be aware of. Having reviewed some of the assumptions that go into estimating p-values and challenges in both data and analytics, you can perhaps appreciate the degree of uncertainty inherent in most p-values reported in various forms, including on volcano plots. In particular, the degree of dependence or independence in the data points very much influences the estimated p-value, and that makes it very challenging to rely upon it. It is very frequent to assume that data points obtained from replicates are independent in estimating p-values, and this assumption is going to inflate the confidence of p-values, and therefore proteins that appear significant, in fact, may not be significant. That might simply be a reflection of having assumed more independence of the data points than is warranted. Another problem with volcano plots is the somewhat arbitrary thresholding, where data points are considered significant above a threshold and insignificant below that threshold. And furthermore, there can be very many proteins that are differentially abundant that are simply not identified as differentially abundant because we did not have enough statistical power. We did not have enough replicates. We did not have enough peptides detected from these peptides. But nonetheless, there can be other methods that can identify their differential abundance that don't require even the computation of p-values for individual proteins. For example, if all ribosomal proteins are just below the interface of significance here shown on this volcano plot, while we may not be able to compute the significance of a significant p-value for an individual ribosomal protein, we might be able to compute very confident estimate that ribosomes as a whole are changing in between the two conditions compared here. And in fact, there are methods that can allow to relax or sidestep the assumptions made in estimating differential protein abundance and give us confident inferences of of related, functionally related sets of proteins that are changing across the studied sets of conditions. And one set of approaches that can do this analysis is known as protein set enrichment analysis. It can be performed very broadly, not just in terms of differential protein abundance, but in terms of any quantity of the proteins of interest, whether that's correlation to other phenotypes or whether that is slopes and so on. And this is going to be the subject of our next lecture. I'm looking forward to it.